clinical samples isolated in 2009 and 2010.
Thank you, Emmy. We have a collaboration with the Botswana Veterinary Institute, and colleague from Botswana Veterinary Institute, Latoya, is here. We have two posters, and this is one poster that I will present. It's about that complete genome sequencing, and the project is supported by two projects, Pediafield and APSEC. And what we have done is to try to do the full genome sequencing directly from the samples. From cattle that had symptoms, we got the samples. The epithelia were taken in Zambia and Namibia and were sent to the Botswana Veterinary Institute and the Botswana Vaccine Institute, and they were highly positive. In the PCRs, they were really, really highly positive. And the poster will guide you through the method that we used. So we do homogenization, filtration, and then the RNA extraction, and then the reverse transcription we did with random hexameres. Then the library was prepared with the Illumina Nexteriac XT kit, and the sequencing itself was done with the Illumina MySeq. What is important that you do the data quality control and that you try to get rid of those nuclear amplicons that has little or no information or very little, so this is the trimming, and then you do the de novo assembly. We had foreseen that if the de novo assembly was not complete, that we could fill in the gaps by the Sanger sequencing. And so that worked well for two of the three strains. The O strain and the SET2 strain worked well. By de novo assembly, we could have the full genome sequencing. For the SET1 strain, we had limited gaps, and we had to close it by the Sanger sequencing. So probably we can also improve the method by using the method that they also do in Perbrite, where they not use the random hexamer primers, but the targeted primers. So this is also something that we will try to do in the future. And in this way, we could demonstrate that it is possible to do full genome sequencing directly from samples from symptomatic animals. And so now we have the full genome sequencing from the OSA Zambia, the SET1 Namibia from 2010, and the SET2 Zambia from 2009. And they were submitted to GeneBank and are now publicly available. So more information is in the poster. Thank you.
Thank you, Chris. There is a time for a couple of questions if there are from the floor.
Maybe it's all very clear.
So you have been very clear.
Okay.
Thank you. So we can move